Hello everyone. Welcome back to Dr. Liu's VCE Biological channel. Today, let's learn together about the 2021 VCE Biology. We will focus on questions related to the regulation of biochemical pathways. My explanations may need to be revised. If any teachers, parents, or classmates find errors, please feel free to provide your guidance. Rubisco is an enzyme found in green plants. Rubisco is important in two reactions. A biology student hypothesized that the same active site on Rubisco is used in reaction 1 and reaction 2. When the temperature is increased, the rates of both reactions increase. As oxygen concentration increases, Rubisco's active sites will be occupied to catalyze reaction 1. So, the rate of reaction 2 will decrease. If the concentration of Rubisco is increased, the rate of both reactions increases, not decreases. Assuming the concentration of carbon dioxide increases, the active sites of Rubisco will be occupied to catalyze reaction 2. So, the rate of reaction 1 will decrease. Scientists studied cells in the embryos of the nematode worm Caenorhabditis elegans. They identified 3 proteins, SED3, SED4, and SED9, that are involved in programmed cell death. When no death signaling molecule was detected by an embryonic cell, SED9 inhibited the action of SED3 and SED4, as shown in diagram 1. When a death signaling molecule was detected, SED9 changed shape and became inactive. SED3 and SED4 were no longer inhibited, and their activity resulted in the changes shown in diagram 2. Within the VCE course, programmed cell death means apoptosis. The cell in diagram 2 is undergoing apoptosis. Since SED3 activates protease and nuclease, it most likely is a caspase. The key word is embryos. So, apoptosis most likely ensures the appropriate formation of developing organs. The diagram below shows a small part of a cross-section of the plasma membrane of a cell. Some substances can move directly through the phospholipid bilayer. Complete the table below by naming one small hydrophilic substance and one small hydrophobic substance that can move through the phospholipid bilayer. Provide an example of a situation when each small substance would move through the phospholipid bilayer. The water or ethanol is hydrophilic. Water diffuses into a plant cell during photosynthesis, or diffuses out of the cell during respiration. Ethanol diffuses out of yeast or bacterial cells that are anaerobically respiring. Carbon dioxide or steroid hormones are hydrophobic. Carbon dioxide diffuses into a photosynthetic plant cell, or diffuses out of the cell that is aerobically respiring. The steroid or steroid hormone diffuses into a cell and binds to a receptor. Some very large substances, and/or large particles, that do not dissolve in the phospholipid bilayer, can still move into or out of a cell. Using one example, explain how the phospholipid bilayer transports these very large substances, and/or large particles, without the use of channel or carrier proteins either into a cell or out of a cell. Select a direction of transport for your response by ticking the boxes. If the student selected into a cell, they needed to name one of the following: endocytosis, phagocytosis, or pinocytosis. The plasma membrane surrounds the large substance, and a vesicle forms around the substance. Some suitable examples are proteins, antigens, viruses, or bacteria. If the student selected out of a cell, they needed to refer to one of the following: exocytosis, bulk transport, or requires ATP. A vesicle containing the large particle fuses with the plasma membrane and the contents are released from the cell. Some suitable examples are proteins named proteoproteins, peptide hormones, neurotransmitters, enzymes, or antibodies. Outline what is meant by the term cell signaling. 2 marks cover 2 points. Transmission of a signal to or within a cell by a signaling molecule. This involves reception and response in the cell receiving the signal. 2 different modes of cell signaling occur over a short distance. In mode X, the signaling molecules are secreted outside cells and then they bind with receptor within the target cell. In mode Y, the signaling molecules are exocytosis and they bind with the receptor on the surface of the target cell. Complete the table below by naming an example of the signaling molecule and target cell for each mode of cell signaling shown in the diagrams. For mode X, the examples of signaling molecules are cytokines, interferons, histamines, death ligands, or hormones. The responding target cells are phagocytes, or T cells or B cells, cells near virus-infected cells, phagocytes, or any appropriate cell types, cells to undergo apoptosis, or relevant target cells. For mode Y, the examples of signaling molecules are neurotransmitters, or neurohormones. The responding target cells are neurons, muscles, glands, neurons, endocrine cells, or immune cells. Cell signaling has been observed in honeybees in nests. If a hive is disturbed, guard bees secrete a substance that smells like bananas from sting glands in their abdomens. This signaling molecule is a pheromone. Guard bees fan their wings to move the pheromone. In the regulation of biochemical pathways, 4 multiple-choice questions are worth 4 marks, and 2 short-answer questions are worth 11 marks. The overall score for this section is 15 marks. With a total possible score of 120 points, the percentage for this section is 12.5% of the total score. If you need my help, please add me on WeChat or WhatsApp and send me an email. Thanks for your attention. Please note Dr. Liu's VCE Physics channel, Dr. Liu's VCE Chemistry channel, and Dr. Liu's VCE Biology channel.